Hi dear students, so here we are starting part 2, the chapter Biomolecule, the most important NCRT based question for the upcoming class 12 chemistry exam. So let's go to the next question, all of you. Here we have a new question. Which of the following represent a peptide chain? You know, a peptide chain we can observe in between two amino acids, right? In between two amino acids we can observe it. Let me give a small example for the connection between two amino acids. Let me take one amino acid is called glycine and let me combine with the alanine. Okay, alanine. You know, glycine means the general formula I told you RCHNH2 with the COOH. If the R group is hydrogen, then it is called which group? Glycine, which are amino acid glycine. If R group is CH3 group, it is called alanine. Alanine. Okay. Let me take the glycine and alanine in small another way. Let me take NH2 group left side. Okay. So NH2. CH2 COOH. All of you check it. This is glycine, right? NH2 CH2 COH plus. What is alanine? Now let me write NH2 group again to the left side. So like I can write NH2 CH CH3 COOH. Correct. This is the one alanine. You know, this is simple, just nothing but elimination of water. Okay, from carboxylic acid. From carboxylic acid you can take the OH group. From NH2 group you take one hydrogen, eliminate as water. Similarly, from or you remember like this, wherever you see NH2 group, take one hydrogen. Wherever you see NH2 group, take one hydrogen. Wherever you see carboxylic acid, take OH group. So what is the left side? You have NH, CH2, C double bond O connected to the other chain NH, CH, CH3, C double bond O. This is the chain. And I told you the bond between two amino acid is a peptide bond. Peptide bond belongs to which type of bond? Whether it is ether, amide or polyester type. Amide linkage. Can you see that? CONH bond. CONH group is said to be which bond? Amide linkage. Okay. So the question, let's see. Which of the following pair represent a peptide linkage? Where you can observe a proper peptide linkage? Option A, you can see NHCO bond is there. After that NHC, but here oxygen is not there. Not a perfect one. Option B, NHCO is there. Then CH2, CH2, CH2. So see, so many groups are coming in between. Then NH. After NH, CO group is not there. NH group nearby CO group will be there. So you see that here NH, CH2, CO, NH is there, and CH2, CO, NH is there. Yeah, this is a perfect manner. Option C is the correct one. But what about here? CO, NH, NH, CO nearby NH2 group is not there, right? CO, NH, that is the master component or amide linkage in peptide linkage. Option C. The peptide linkage formed between glycine and alanine. Yeah, I just explained before which is glycine and alanine, dear student. Glycine and alanine combined with this one. This is called glyala. Glyala. Funny, right? Glyala. Glycine and alanine combined together, and the compound is glyala. That is a protein called. Can you identify this chain anywhere? NH, CH2, CO, NH, CH, CH3, CO. Or you can find here NH2. Okay, they didn't eliminated completely. No need to eliminate completely at the terminal because it's only two things are there, right? Only two things are there. So no need to eliminate from the end. If it is so many compounds comes, you have to do like that. So NH2 COOH. This one you can keep it. So NH2 CH2, NH2 CH2 CONH. CH3, COOH. You can see observed in option B, I think. Okay, check it there. NH2, CH2, CONH. Yeah, option B is the correct answer. Okay, next one. In fibrous protein, polypeptide chains are held together by. There is a simple definition given in textbook in fibrous protein. In fibrous protein, the polypeptide chains are running parallel. The polypeptide chains are running parallel and they are held together by strong hydrogen bonding. Therefore, they are little stiffened one. They are insoluble in water. So, hydrogen bond is the bond between two fibrous protein, polypeptide chain. Globular proteins are present in globular protein. You know, globular protein example we studied insulin and albumin. They occurs in blood, eggs, milk, right? All of these, all are present there. Primary structure of protein we discussed, it is the specific sequential arrangement of amino acid on a polypeptide chain. So sequence in which alpha amino acids are linked to one another. Yes, correct. Option A is the correct answer. Most common type of secondary structure. Secondary structure we classify into two, alpha helix and beta pleated. Alpha helix and beta pleated. See option A, alpha helix and beta helix. No, one is helical structure, one is sheet-like structure. Alpha helix and beta pleated. Option B is the correct one. Which type of interaction are responsible for making the alpha helix structure stable? Here all, if you consider a helical structure of alpha helix structure, you know, if you consider the amino acid as like R, CH, NH2, COOH, I told you when amino acid combine together or coming together, from NH2 group you have to take which one? NH2 group. So what is left? NH. From carboxylic acid you take COH group. So what is left? CO group. Correct. Correct. Such a kind of so many groups will be there in each turn. So in one turn there will be a CO group. In another turn there will be a NH2 group. You know how they are coiling? That is the question. You know, polypeptide chain is a linear one. How they are coiling in alpha helix? In one strand, in one turn, one NH group from one amino acid. In another turn, CO group in another amino acid are held together by hydrogen bond. So when they come closer, the linear polypeptide chain coil. This is what happened. So which type of interaction are responsible for making the alpha helix structure is one. So what we can say, it is not a peptide bond, it is a hydrogen bond between NH of amino acid in one turn with the CO amino acid of adjacent turn. Yes, option B is the responsibility for the alpha helix structure of Denaturation of protein leads to the loss of. Dear student, let me tell you what is denaturation. Some more question we can discuss about that. Most expecting past, denaturation of protein. You know very well, a protein when it is normal stage with a perfect biological activity is known as native protein. Native protein, with a perfect structure, perfect geometry, perfect function, everything. When a native protein subjected to a change in temperature, that is called physical change. When you make a change in pH, that is called chemical change. So generally, when a native protein subjected to a physical change or chemical change, what will happen? You know, then denaturation takes place. During denaturation, what all things are happening? Dear all, keep in mind, you know, secondary and tertiary structure disturbed. Tertiary structure completely destroyed, but primary structure remain the same. Primary structures remain intact. Nothing changes. Another thing, its biological activity loss. Another one, during heating or making pH, the globular protein, get unfold. and also the fibrous protein that is uncoiled. Okay, uncoiled. So these all things are happening during denaturation. What are secondary, tertiary structure destroyed completely, primary structure nothing happened, biological activity loses, globular protein get unfold, means globular protein become fibrous protein, and fibrous protein that is completely uncoiled, try to cut uncoiled. So denaturation of protein leads to loss of its biological activity by the loss of both primary and secondary, no, loss of both secondary and tertiary structure. Its loss is biological activity because secondary and tertiary structure is completely disturbing. Mark the wrong statement about denaturation of protein. The wrong statement here on. Primary structure of the protein does not change. Correct. Globular proteins are converted to fibrous protein. Correct. Fibrous proteins are converted to globular. No, when you heat the protein, the fibrous mean it is a fiber like one. Globular mean a spherical one. When you heat it, the spherical globular get unfold and it become a type of fibrous. Not fibrous to globular. That is wrong. Biological activity of the protein destroyed. Correct. Option C is the answer. On boiling egg, boiling egg and curdling of milk is an example of denaturation. What structural changes are taking place in the egg white? The color of the egg changes from question, not the color, the structure. Secondary and tertiary structure are destroyed, but primary structure remain intact. Correct, right? Yes. On boiling egg, what structural changes takes place? Secondary structure completely disturbed, primary structure remain the same. Next, nucleic acids are, nucleic acid, DNA and RNA are composed of so many number of which one? Nucleotides, long chain polymer of nucleotides. Which of the following is not produced by human body? Whether it is enzyme, protein, vitamin, nucleic acid. The human body cannot prepare which one? Vitamins. Nucleoside on hydrolysis give. Dear student, what all the things are present in a nucleoside? Part 1 video we discussed detail. Nucleoside which contain a pentose sugar with the first position a base. Right? There is a pentose sugar on the first position connected to a base. It is called what? Nucleoside. So nucleoside on hydrolysis will give you a aldopentose and nitrogenous base. Yeah, aldopentose and nitrogenous base. Guanine is an example of guanine. Guanine is an example of, dear student, guanine, adenine, thymine, guanine, cytosine, uracil. Guanine is a nitrogen containing base, right? Nitrogenous base. Option A is the answer. In DNA, the complementary bases are, we studied in DNA, if one strand contains adenine, other strand is connecting with the thymine with a double bond. One strand contains guanine, other strand cytosine through a triple bond. So adenine, thymine, guanine, cytosine, that is the pair. Adenine and thymine, guanine and cytosine. Option B. RNA is a, come on, RNA is a, which type of strand, dear all of you? RNA is a single stranded one, right? RNA, the DNA, the major difference. RNA having a single strand, DNA having a double strand, right? Double strand. In one strand of DNA as this sequence, okay, DNA, let me take one strand. In one strand they contain, what are the species, what are the bases? Adenine, thymine, guanine, cytosine, thymine, guanine, adenine. Correct. So what is the sequence of complementary strand? What will be the other strand? Adenine always with which one? Thymine. Thymine always with the adenine. Guanine with the cytosine. Cytosine with the guanine. Thymine with the adenine. Thymine with the adenine. Guanine with the cytosine. A with the thymine. So can you identify this pair anywhere? TACGAACT. TACGAACT. Option D, right? Yes. Match column 1 with column 2. Nucleoside, dear all. What all things are present in a nucleoside? Nucleoside which contain a pentose sugar with a base. A pentose sugar with a base. A with the third one. A with the third one. Only one answer. Option A. Let's check others also. Nucleotide which contain a pentose sugar, a nitrogen base with a phosphate group. Right? Phosphoric acid group. That is with the first one. DNA contain the cytosine and thymine. DNA contain thymine, RNA contain uracil. Right? Option A is the correct one. Match column 1 with column 2. Again, pentose sugar in DNA. Which is the pentose sugar in DNA? Or what can be matched with that? Pentose sugar in DNA. DNA pentose sugar we discussed, no? Pentose sugar like this. Pentose sugar in DNA, it is a pentose, right? It is a furanose ring. Furanose ring. 4 carbon on the chain and 1 oxygen. Furanose ring. So pentose sugar we can match with the 4th one. Nucleic acid. Nucleic acid. Anything is matching? Nucleic acid. Nucleic acid is DNA, RNA. They are the genetic material. RNA contain uracil. Yes. And vitamin we discussed, no? Ascorbic acid. Ascorbic acid is vitamin C. Yeah. So all together we can match with option A is the correct answer. Okay, dear all, next one. The two main difference between RNA and the DNA are. What is the major difference between RNA and DNA? Option A, ribose sugar and thymine in RNA. Is thymine present in RNA? No. Deoxyribose sugar and uracil in DNA. Uracil is present in DNA? No, it is in RNA. Ribose sugar and uracil in RNA. Yeah. Major difference between RNA and DNA are pentose sugar, there is a difference. We discussed in part 1. RNA which contain the pentose sugar called beta-D ribose. In DNA it is called beta-D 2-deoxyribose. And RNA contain uracil, DNA contain thymine. Yes, that is the difference. Option C is the correct one. RNA molecules are of 3 types, you know. The 3 types of RNA we discussed at the end of the chapter. They are said to be mRNA. What is mRNA? Messenger RNA, tRNA and rRNA. Messenger RNA, then transfer RNA and also the ribosomal RNA, MTR, messenger ribosomal transfer RNA. Option C is the answer. The double strand helix structure of DNA was proposed by, you know, the double strand of, we discussed one question before in part 1, that is Watson and Crick. Watson and Crick. Consider the compound. You can see there is a compound. Arrange X, Y, Z in the order of increasing acid strength. Carefully all of you listen. This is X, Z, Y. Dear all, question is increasing order of acid strength. Can you tell me which group is highly acidic in nature? No doubt. Which one? Carboxylic acid. Correct. X is greater acidic in nature. So we can cancel option B and D. Either A and C. X is very much greater because it is carboxylic acid. Now the question, why NH3 plus actually acidic? I told you in Zwitter ionic form, in NH3 plus ion, it can lose one hydrogen and convert it to which one? NH2. Stable state. Correct. So they are acidic in nature because they are ready to lose. Dear all, you know, one NH3 plus group means this NH3 plus group is very near to the carboxylic acid. This NH3 plus group is away from carboxylic acid. You know, carboxylic acid is a electron withdrawing group. Carboxylic acid is a electron withdrawing group. We studied in alcohol chapter. Electron withdrawing group can increase the acidity. So if there is an electron withdrawing group present nearby the NH3 plus, from this compound you can remove H plus so fast. So the Z1 will be more acidic and the weak one is Y. X, Z, Y. Answer is option A. Okay. Then, which of the following statement is not true about glucose? Not true. It is an aldohexose, correct? It contains one aldehyde, 6 carbon, correct one. On heating with a hydrogen iodide, yes, it will form normal hexane. It is present in furanose form. No, glucose we are expressing in pyranose form, correct? Pyranose form. Which one we are expressing in furanose form? Fructose, correct? And it does not give 2,4-DNP test, dinitrophenyl hydrazine test, Bosch's test. Okay, just remember only the name. It does not give 2,4-DNP test. Yes, that's a correct one. Okay, dear all. So in biomolecule chapter, nearby around 100, just nearby 100 questions we are discussing as a part 1 and part 2 video. All of you make sure that just as per MCQ exam, as per the new pattern, it's very important to know the basic concepts or the just theory for the chapter biomolecule. Direct question you can expect. So keep practicing or keep reading the NCERT textbook line by line, and please keep practicing the all question which we discussed here. We can expect the same question in your upcoming 2021 exam paper. Okay, so wish you all the very best and practice well. We will meet again with other chapter video in the coming days. Thank you all.